Good afternoon and welcome to Te Whare Wanaka o Aotearoa, Lincoln University. This is the next event in the State of the Land Bioeconomy Series, which we're really proud to co-host with our colleagues at the Bioeconomy Science Institute. As Katie said, I'm Chad Hewitt. I'm the provost of the university. The Vice Chancellor gives his apologies. He's away today. Usually he announces these events and MCs them. The State of the Land Bioeconomy Series is delivered, as I said, in partnership with BSI, bringing together researchers, industry leaders, policymakers, and communities to explore the opportunities and challenges shaping Aotearoa New Zealand's land-based sectors. Through these conversations, we aim to strengthen the connections between science, innovation, and practice, ensuring research contributes to informed decision-making and supports a more sustainable, productive, and resilient future for our land-based systems. Today's session explores the transformative potential of microbial fermentation, a field of science that is increasingly shaping the future of food production, sustainable agriculture, and the wider bioeconomy. While fermentation has a long history in human food systems and drink, advances in microbiology, biotechnology, and production methods are opening new opportunities. Microbial processes are helping us explore new ways to improve resource efficiency, reduce waste, develop high-value products, and support a more circular approach to production. For New Zealand, these innovations present exciting opportunities across our food, fiber, and agricultural sectors. They also highlight the importance of collaboration between researchers, industry, government, and communities to ensure scientific discoveries can be translated into practical solutions. Today's speakers will share insights from two complementary perspectives, exploring microbial processes in food and beverage systems, as well as applications in sustainable agriculture, including bioprotection and enhanced plant production. Before we begin, I'll briefly outline the format of today's session. I'll introduce both of our speakers at the same time. Each of them will give a 15-minute presentation sharing their research and perspectives on microbial fermentation. We will then move into a moderated discussion with both speakers, followed by an opportunity for audience to ask questions. We aim to conclude the formal presentation at 2:00, after which we invite you to the foyer for light afternoon tea, as Katie indicated. We're fortunate to be joined by two highly respected researchers working at the forefront of microbial science and its applications. Our first speaker is Professor Stephen On from Lincoln University. Stephen is a professor in microbiology with research spanning microbiological food safety, microbial processes in winemaking, diagnostic methods, and bacterial taxonomy. His work explores how understanding microorganisms can contribute to food quality and safety, public health, and innovation within food and beverage systems. With more than 30 years experience in research, Stephen has published extensively in microbiology and has contributed his expertise to industry, government policy, and food safety investigations. His current research includes microbial characterization, food quality and safety technologies, antibiotic resistance, and the application of microbial knowledge to support innovation in agricultural and food systems. Joining him is Dr. Laura Villamizar from the Bioeconomy Science Institute. Yes? Okay. Laura brings 3 decades of experience in biological control, microbial production, fermentation, formulation development, and sustainable agricultural biotechnology. Her research is focused on the development and application of microbial products, including biopesticides and biofertilizers, using microorganisms as entomopathogenic fungi, bacteria, and baculoviruses. Her expertise spans microbial identification, production systems, quality control, field evaluation, and the pathway from research discovery through to commercial application. Her work has contributed to the development and regulatory approval of innovative microbial solutions for sustainable agriculture, across Colombia, Brazil, and New Zealand. So together, Stephen and Laura will bring complementary perspectives on the role of microbial fermentation in the bioeconomy, from food innovation and microbial processes through to sustainable agricultural applications. So please join me in welcoming Professor Stephen On and Dr. Laura Villamizar.
Kia ora, everyone. Thank you, Chad, for that introduction. It's so nice to see so many of you here today for this seminar. With apologies to the organisers, I thought that this was a better title than the rather more formal description that was given. Fermentation is bigger on the inside, because it is. And to explain this perspective, I want to ask you a question first of all. How many people here are fans of Doctor Who, the TV series? Yes, you're already halfway there, probably. So, to explain to those who haven't watched Doctor Who, it is the world's longest running science fiction TV series, started in 1963, and it involves the adventures of an intergalactic, interdimensional, time-traveling person that has these wild adventures. And this is what he travels in. It's an old-fashioned British police box. Doesn't look very slash, does it? The thing is, him and all of his companions, as soon as they step through those doors, it's bigger on the inside. The TARDIS has hundreds of rooms strewn through its interior, filled with all sorts of different stuff, and that's why it's just like fermentation. What are fermented products? I'm sure you can all name quite a few, right? Wine, beer, cider, kombucha. What about yogurt, cheese, bread, and some types of fermented fish? They're all fermented. But wait, there's more. Vinegar, chocolates, and coffee. Each of those products have an element of fermentation actually at their core. They all rely on fermentation at some point in their production. Oh, but wait, there's more. Probiotics are products of fermentation. You know the coating on sugar-free mints? That's a product of fermentation. This is a picture of a plant in Japan used to produce glutamate. And the reason that it's so big, there are 7 fermenters in that picture, each of them 230 square meters large. Why are they so big? Because monosodium glutamate is used in just about every processed food that we actually consume. And that's the thing about fermentation. It's actually a very diverse science. It's no one thing. Types of fermentation include alcoholic fermentation, lactic acid fermentation for cheese and yogurt, acetic acid fermentation, and last but not least, and it's used as a sort of catch-all term by which we just grow microbes up and either use them directly or extract what we want and apply that to. And that's fermentation. One term, multiple sciences. Now, Laura is going to talk to you predominantly about the 4th example. I'm just going to talk about booze. So sometimes I present to the wine industry, and this description sort of to give you potentially annoys them just a little bit, but just to say that anyone in this room can produce wine. Anyone. All you need is grapes. You just press the grapes, get the juice, and leave it. And at some point, it will turn into wine. You can do it here. You can do it in your living room. You can do it in your garden, as long as it's sort of covered. And the reason that we can do it basically with no added intervention at all is because grapes inherently have every organism that you actually need to produce wine. as well as in the atmosphere as well, right? So there's no barrier in terms of as long as you've got your substrate, it will turn into wine. And I think probably in the last 20 years, because our methods of detection of different microbial species, they've got much more advanced. We have things like the Bioeconomy metagenomics that gives us potential to detect basically everything that is in and on the grape surface, soils, atmosphere, you name it, we can do it. And so that is some of the insights that we've actually had. Some of the stuff that has been uncovered over the years with the application of metagenomics and metabolic coding is effectively a far better understanding of the role that microbes have in terms of determining the type of wine and the type of style of wine and its uniqueness, if you like, as opposed to just understanding that it's a grape varietal. It's where your grapes are grown in terms of soil quality, in terms of temperature and all this stuff. This is a slide that I use in teaching. It's 2015. A study that basically shows, so this metabolic coding basically shows you each different colour is a different microbial, in this case yeast, a different yeast genus. So every time you see a different colour, it's a different class of organism, right? That slide shows you samples of grapes from different regions from Spain, Italy, and Argentina. And if you don't even have to look terribly closely, right? Every bar is different. And so what we've come to understand is that the uniqueness of a microbial population in terms of a region actually has a distinctiveness all of its own right. What about New Zealand? So I want to show you this picture that we were very, very fortunate to be able to work with a unique winemaker in this country, Greystone Wines. They produce all of their wines through spontaneous fermentation. So they don't add, in the sort of classical fermentation sense, they don't take a single strain or bung it into the grape juice. They just let everything happen naturally, okay? So what you see there is a subset of microbial populations. The other thing that's unique about Greystone is that they take their grapes and they ferment them in two different places, so to speak. Inside the winery and in the vineyard, they have these tanks basically outside. So indoors and outdoors. And what you can see on that side, again, different colours, different, in this case, bacterial groups, different genera. And you can see there that it's actually, bearing in mind that that's actually the same bunch of grapes, just fermented indoors and out, the colours are different, right? So you actually have, you're seeing the influence of the environment on the proliferation of organisms right from the start. What about this? Does that look strikingly different to you? Yes. Thank you. It's the same place. The top one's the vineyard, the second one's the winery. Difference, 2018 and 2021. That is a massive shift in terms of your microbial population, just in the space of 3 years. We looked at climatic differences, publicly available, and what you can basically see is quite big shifts in temperature from one year to the other, quite big shifts in rainfall from one year to the other. I think if ever there was an indication that climate change has huge potential to shift the microbiota in a wine production facility, it's most certainly here. Does it make any difference? Well, we got real lucky with this. We actually had bottles of wine that had been made from those different tanks, basically, indoors and out. And we managed to do basically some taste testing. What you can see here is kind of non-metric plot, if you like. Can you see the kind of the purple and the green? In terms of blind taste testing, you're seeing a shift in terms of how people perceive the flavours from wines that are produced from grapes indoors and out. So it's having an influence on how people perceive the wines. What might that mean longer term? It shouldn't actually be a surprise. This is another slide that I use in teaching. Very small font. I only want to focus really on the top one, which I can read to you quite easily. But wine is a product of fermentation, and the yeasts and the other microorganisms that basically are present that do the job. Their metabolic properties mean that they're turning sugars into alcohol. Sure, they're also producing a lot of other things. And I like this slide because if you just take a look at the top one, ethyl acetate. So it confers a nail polish, a kind of fruity aroma in terms of your wines, right? If you take a look at the concentration of wine summarized from a number of studies, it goes from 22.5 to 63.5. That is likely to be, at least in part, the difference between different strains. Different strains of yeast you're actually using because they produce this stuff at a different rate. So microbes have a significant influence on, frankly, whether or not it's plonk you're drinking, or whether or not you're going to win a gold medal. So that's part of the explanation why different yeasts make different wines. We wondered, could that potential be predicted? So this is some work I did with BSI, AgriSearch as there was, using a technique called Māori mass spectroscopy, or matrix-associated laser desorption ionisation time of light mass spectrometry. Just to summarise, you basically have a sample, you blast it apart with a laser beam, you separate the component fragments on the basis of either their mass or their charge, or both. You detect that. And in terms of yeast characterization or anything really, you end up with these different types of spectra. So you can take these spectra and you can use, fancy algorithms and computers to sort of do different things. Basically calculate similarities between individual strains and of course groups when you've got populations. So we've done this. And this is where we kind of at least start against it. First of all, this is a study published 3 years ago, I think, where we could see, using different types of, again, sort of a modeling software, you can quite easily see a difference between strains used in brewing compared to strains used for winemaking. So if you had a completely unknown strain that you've just isolated, what's it good for? This is arguably one of the things that you could do to predict its use. This is what happens when we have a bigger population of the same population, but this is the wine strains only. And so with this, again, a different kind of modeling approach. Let me talk you through this. So that group 2, that's a group that is dominated by strains only used to make a Prix de Meaux. Group 1, predominated by strains that are used to make white wine. Both of those two groups have certain traits in common. They are better at fermenting at low temperatures, and they produce higher alcohol levels than group 3, 4, and 5. Groups 3 and 4, again, used to make red wines. Group 5, almost exclusively white wine making strains. So these are all commercial strains that are available. But already you can see this kind of grouping has relevance to how they are actually used in industry. So again, if you had indigenous strains, well, you could have some sort of an idea. We've done something similar for beer, and arguably the separation is even nicer. In actual fact, a nice grouping of lager making strains with ale strains and cider strains. And I mean, again, sometimes you just have to use a different analytical approach to ask the right questions to get the result that you want. And here you can see a strain used to make low alcohol products. Low alcohol beers and wines are becoming very popular in this day and age, and that readily separates out from the rest of the strain. So quite a useful method, I think. I'm almost finished, and this is in many ways my kind of favourite part of the talk. It's very, very new. We discovered something, which I'm only going to call organism X. And just curiosity-driven research, wasn't looking for it. Not you don't go out to explore absolutely everything. So this is what it is. It's a bacterium. It's not a yeast. It's not anything that is traditionally involved with wine making, like a lactic acid bacterium. Some species can be used for malolactic fermentation. Acetic acid bacteria do exactly what it says on the can. They can turn wine into vinegar, so not so nice. But this organism isn't anything like that. It's ethanol tolerant, it's completely natural, and I believe, that it has huge potential for flavour improvement in white beers, ciders, but actually entirely new products. Why do I say that? Because just looking at what it actually does, basically in juice, it produces an extensive array of pleasant flavour and aroma chemicals, very wide range. And having been one of the people that sort of stood in a lab and basically smelt the results of what happens when you drop it into juice, it's quite spectacular. So I'm very excited about this. Throughout my career, I've been really well served with very positive collaborations. So this is just the list. I've also had some excellent PhD students whose work has laid the foundation for all of this. So John and Pruth is a current PhD student. Agogo is a former PhD student. So John, Ben was instrumental when I came to Lincoln to help me make these links with and get involved with basically wine making research. Chris is an expert molecular biologist. Richard is an excellent biochemistry technician. I've had the privilege of working with people from Ag Research, Jeff and Stefan, as well as Mandarin at Lincoln Research. Thanks to Don Maxwell at Greystone Wines, basically allowing us to collaborate and some funding from both Lincoln University and KiwiNet. And that's my presentation. Thank you. And shall I pass over to Laura? Now, please take the stand. I should pass the baton.
Thank you, Stephen.
Thanks. That's forward.
Well, good afternoon, everybody. And after this great overview of fermentation, I'm going to discuss how fermentation can be the bridge between having a beneficial microbe and being able to use it as a microbial solution for agriculture. Microbial solutions for agriculture or microbial products are basically microorganisms or microbial metabolites or any kind of product that is derived from a microbe that can be applied to plants, to soil, or to seeds in order to control diseases, pests, remediate soils, enhance nutrition, productivity, and resilience. The market for this product has been growing very, very fast during the last decades and during the last years, and this has been driven by different factors. One is consumer demands, because consumers want products that are cleaner, they don't have a lot of chemical residues, so they are pushing the market for more biological solutions. There is also climate change. We need solutions to be more resilient to all the change in our weather and our climate. There is also the need to repair degraded soils, and there is tighter chemical regulations and a lot of agrochemicals that are removed from the market and will be removed in the coming years. The market for microbial solutions is expected to be worth around $27.9 billion in 2028. So you can see the size of the market and the huge opportunity in this space. There are different kinds of microbial solutions for agriculture. The three main products are biopesticides, which are products that suppress or control plant pathogens, insect pests, and weeds. Then we have biofertilizers, products that improve nutrient availability and uptake. And biostimulants, products that stimulate plant growth using multiple direct and indirect mechanisms. But to be able to move a microbe from being just a beneficial microbe to a commercial product, we need to follow a systematic sequence of steps that needs a lot of time and a lot of investment. I have captured the 6 main steps to make this possible. And the first one is discovery. Discovery is when we go to the field, we find the insects, plants, we collect samples, bring to the lab, isolate microbes, characterize them, identify them. Then we have to do screening, and then we find some candidates that have potential to be developed as a product. Then we need to move to another step, which I call design. I think this is a very important step in the development. Is where we define what are we going to use as active ingredient. Is the biomass, is a bioactive molecule? How are we going to produce it? What is the mode of action? Where is our target? How we think our product needs to be delivered. So because this will give us an idea, we need to develop a liquid or a solid or a granule. Everything needs to be analyzed at this point to have an ideal product. And something that is also very important in this step is to do a little bit of market analysis. It's important to know if there is market for this product, if there are competitors in the market, and also how much it's going to cost to produce it. Then we can start the real development. And the next step, development, has 2 main components. The first one is fermentation, and the second one is formulation. Fermentation is in order to be able to produce enough biomass or enough bioactive molecules. So we need to develop a system to produce this active ingredient. When we have this system, we can develop the formulation. And the formulation step, the aim is to transform this active ingredient in a product that is stable, is easy to handle, and is easy for farmers to apply. When we have some prototypes for these formulations, we can start the validation. And validation is doing all the testing of efficacy under laboratory conditions, glass house or field testing, and also determining the shelf life. This happens for many prototypes, and we are filtering the best ones. And when there is this product that is matching all of these characteristics that we define during our design of the product, this is the product that can move to the next steps, registration, and finally commercialization. As you saw, fermentation is a very important step in the development. And for microbial products for agriculture, there are 2 main fermentation systems that are used: liquid fermentation and solid state fermentation. Liquid fermentation basically uses a liquid medium that is normally very simple, that is very defined. As equipment, there are fermenters, and the availability of fermenters is very wide. There are many suppliers with many sizes, from liters to thousands of liters, in which you can control pretty much every condition for the process. This fermentation is normally used for bacteria and yeast. The cost of production is very cheap, but it's important to know that this fermentation requires high capital investment because you need to set up all of these fermenters and all of these facilities for production. The other system is solid fermentation. And in this case, the substrate is not liquid, it's solid. What is normally used for this fermentation is normally cereal grains like rice or sorghum or wheat or barley. In some cases, agricultural waste, or in some cases, kind of coarse like zeolite or pumice that are just supplemented with nutrients. There are fermenters for solid fermentation in the market, but the availability is not similar to liquid fermenters. It's very limited. Fermenters for solid fermentation are very, very expensive. They don't allow to manage very big volumes, so it's not very common to be used. Most of the companies in the world, they use a very simple system, as you can see in the picture, and it's basically putting the substrate in bags, putting in the autoclave, inoculating, and then just maintain them in a room with shelves for incubation. It's very simple, but it works. The problem with this system is it's very labor demanding. You can imagine people just filling bags and inoculating. It takes a lot of labor. And it also produces a large volume of solid waste. And the conditions are not as controllable as you can do in a liquid fermenter. This system is mostly used for production of fungi, could be mushrooms, or could be in the agricultural applications molds. But anyway, fermentation plays a key role in the development of products for agriculture because fermentation can determine biomass quality and robustness. So if we have very healthy biomass, it also determines yield and productivity, how much we can produce, scalability, are we able to scale up the process to produce in large volumes, cost of production, batch consistency, we have repetitivity between batches, and commercial feasibility, which is the ability to manufacture enough product at a competitive cost. I would like to highlight stability because in the development of microbial products for agriculture, stability and shelf life is one of the main constraints. From all the attempts to develop a microbial product for agriculture, only 10% can reach the commercial, the commercialization, basically. And this is for many factors, but one of the factors is shelf life. The shelf life is very short, mainly the products which the active ingredient is the biomass, because we are talking about alive microbes, alive organisms that are susceptible to conditions like UV radiation, high temperature, desiccation, or competition with other microbes. But if we tailor the fermentation conditions, we can help microbes to survive better. So if we play with the composition of the culture medium and all the fermentation conditions like temperature, oxygen, time, we can have biomass that has enhanced stress tolerance. Then we have longer shelf life, better field performance, and then we are improving the commercial viability. This is what we have been working in this platform that we have been developing in the Bioeconomy Science Institute of New Zealand. And this platform has been focused in developing microsclerotia production for beneficial fungi. Microsclerotia are small aggregates of mycelium that are melanized and are very tolerant to abiotic stresses. They are normally produced by plant pathogens to survive for many years in the field, but they are not naturally produced by most of the beneficial fungi. But it's possible to force beneficial microbes to form this propagule using artificial induction with specific conditions in liquid fermentation. So we can produce a propagule that is tolerant to a lot of environmental conditions, but we are also moving from solid fermentation that have a lot of problems for production to liquid fermentation that is easy to scale up and is less labor demanding. We have developed this system for different fungi. I have some examples in this slide, like Aotearoa New Zealand, and Metarhizium robersi. They are all entomopathogenic fungi active against important pests like black beetle, porina, and whitegrass. The full development of a microbial product can take a lot of time. For biofertilizers and biostimulants, possibly less, because in most of the countries, they don't need registration. But biopesticides is pretty much in every country. It's compulsory to have a commercial registration, and it can take several years. As you can see in the map, for US and Brazil and China, it is estimated in approximately 2 years, but for Europe, it's approximately 7 years. So the full development from discovery to commercialization has been estimated in approximately 10 to 15 years. The investment is also very high, has been estimated in up to $10 million for the full development. So it's very expensive. However, there are alternatives for some countries where there is not enough funding to do this development, or there are some regions where people cannot access these products from overseas, or they are too isolated, or there's no enough funding to get them. So there is an alternative to that, and it's what is called on-farm production. And on-farm production relies only in fermentation. This system consists in production of beneficial microbes by farmers or companies for their own use. In this case, there is no need of registration because they are not going to commercialize. They produce, and they use by themselves. Some of the advantages for these systems is they don't need sophisticated equipment. You can see the picture at the top is just a fermenter. It's basically homemade equipment, and you can build a fermenter just with some tanks and having some conditions to keep the aseptic system. There are other systems like the solid fermentation. You only need a room with shelves and good conditions of temperature and humidity. Another advantage of this system is the product can be used very fresh, and possibly this product is more active than a product that has been in the shelf for several months. But there are also challenges in this production. One is contamination, because for the same reason they don't have a very sophisticated infrastructure. It could be some risk of contamination. Inconsistent quality, because the infrastructure is not the best, but also because sometimes people develop these systems is not well trained to do this microbiology work. And shelf life, because products that are manufactured like this, they are not formulated, they are not stabilized, so you cannot really store them for a long time. This system, even these challenges, has been very successful in many places in the world. It was very successful in Colombia to produce Borrelia baciana to control the COVID border. It is actually very, very successful in Africa, in many countries, to produce Fusarium oxysporum to control Striga, which is one of the main weeds in Africa. And we have been also very successful as Bioeconomy Science Institute of New Zealand, working with this system together with our neighbors in the Pacific. They have a pest that is called the coconut rhinoceros beetle, and this pest is threatening the production of oil palm and coconut in several countries in the Pacific. And some of these countries, like we are working particularly with Solomon Islands, Vanuatu, Samoa, and Papua New Guinea, they don't have access to these commercial products, and they don't have companies that can produce them in country. So we have been supporting them with a project funded by MFAD to develop their own on-farm production. We developed an inoculation system. We provide them with a very pure inoculum and some tools so they can set up the substrate and inoculate in very aseptic conditions. Then we help them to set up a room for incubation, very simple, just shelves and some temperature control. And we also provide them with very detailed protocols and training. So they know how to do the inoculation, but they also know how to follow the quality of the fermented product. So this is very successful. Now Solomon Islands is self-sufficient to produce. Vanuatu is very close to be self-sufficient, and now we are concentrated in Papua New Guinea. And this is my presentation of the application of fermentation for agriculture, and my final messages is, well, fermentation, as you can see, is the foundation for successful microbial products in agriculture, and fermentation can be as simple or as sophisticated as you need, but it is very, very important to be successful in this space. Thank you very much.
Thank you, Stephen. We invite you up. I've got a few questions that have been, I believe, shared with you in advance so that we can have both of your views. We've got 15 minutes. I'd like to save about 5 of those so that we can get questions from the audience at the least. Yeah, please. So the first question is, what has changed to make fermentation such an important bioeconomy opportunity right now? So Laura?
I think we have now a better understanding of microbes. There are, oh, sorry. I think that we have a better understanding of microbes and physiology and how they behave with all of these new technologies and proteomics and genomics, metabolomics. So we can really use them better to produce products for better economy.
Yeah, I tend to agree with that. I think it's an awareness. We become more aware of what microbes can do, what their role is in the environment. I mean, obviously we've been using it in fermented products for millennia actually, but that understanding of specifics of the role of individual strains and individual types, I think that's vastly increased. Could still be better, but I think we've made a lot of progress over the last few years.
So the second question, can you identify common opportunities across food, agriculture, and bioprotection applications?
You want to pick what we name one right now? I did so Saccharomyces, right? So this is the main driver for alcoholic fermentation to make alcoholic products. Interestingly, so it will be present on the surface of grapes as well as the associated roots. Interestingly, it is also associated with soil improvements. So there's a relationship there, and I think that this microbes, they exist. They exist as communities, and quite often they have different ways of staying there, which can do one of two things. And there are some microbes that are able to promote plant growth, as well as limit the incursion of specific pathogens. So certainly the fact that you have microbes with that simplest state of being, this is how they exist, this is how they live. And so once you've actually come to an understanding of what they're doing and where they are and why they're there, and then start investigating their properties, then you've got the opportunity to look at these joint these synergistic applications, because they're going to be there. It's just a question of, it's that discovery part that's the hard part. But once you've got some sort of an idea, and these metagenomic techniques are very useful for getting an understanding of the rawness. I think that has the potential to make great inroads.
I think there are many common opportunities between sectors, because whatever you develop for agriculture, it can be maybe applied for food or for any of the other industries. And it's why now the focus is not only developing products, it's more developing platforms. So you develop this platform to do this with microbes that can be used for agriculture, but then you can use the same platform to produce microbes for food. So this can be shared between all the industries.
So what are the biggest opportunities for New Zealand in the next, say, decade?
I think the main opportunities are not becoming the fermentation center for the world, because we cannot do that. But I think if we can specialize in producing very differentiated products or microbes that are not in any other place, and I think we have a very unique biodiversity. So if we can explore this to produce unique molecules or unique microbes, we will be at the top.
Yeah, I mean, it's well known that New Zealand is host to a wide range of basically unique flora, right? Unique fauna, unique flora. And there are a number of studies out there that basically showing that some microorganisms we have here are also very, very distinct. I think right from the start, if we understand what they're actually doing, that gives us competitive advantage because no one else has them. The discovery part remains the hardest. Just making the point that the wine industry here, as well as an expanding craft beer industry, right? That's over $3 billion a year in terms of just exports, and we mustn't lose sight of that. That's an opportunity. The sales have tended to go up year on year. Even I think the only hiccup was basically when during COVID, when no one went out and no one bought anything, right? But it's an expanding agricultural sector still is. That opportunity to sort of hone one's products, because that's part of the key for growth is to continue to develop your products to go into new markets. Maybe certain regions have a preference for certain styles. That's not going to go away. So we have to continue to innovate those particular products too.
And one last question for me. You've identified discovery as being a critical step. What other barriers need to be overcome to make fermentation front and center?
From the microbial products for agriculture point of view, the main barrier in New Zealand has been infrastructure for production. It has been difficult to find facilities to do fermentation in big scale. Many companies have to go overseas to produce overseas and bring the biomass. So having capability to produce large volumes in country will be the solution for this barrier.
Mum, I mean, you can't have biodiscovery without funding the curiosity-driven research that underpins what's this doing here. Doesn't matter what area of research you're in, whether or not it's human health, plant health, animal health, production of alcoholic products or non-alcoholic products. It doesn't matter. That discovery part is critical, because without that you don't have anything. Who funds it? And to be fair, it's probably not absolutely exclusive to this country, but it's certainly not. It's not a small factor either. To some extent, what was also described in terms of the infrastructure, the plants that are required to. Oh, we've got this great thing. Let's put it out. Well, here we go. That's money as well. So a lot of this stuff actually hinges on getting appropriate investment at the right time to get things moving.
Thank you. Now, I'll open it up to the audience. Just ask, state your name and where you're from, and then ask the question. And if it's oriented towards Stephen or Laura, please indicate that. So, you've stunned the audience. Or scared them.
Oh yeah, Australia, BSI. I'll start. Where are we at in New Zealand in terms of designing microbes to make products, specific products for us? I don't know who that's best aimed at there, but
I can have a stab. I suppose part of this lies in this case, I don't know the specifics, that the idea of gene editing, where you can at least take out and insert genes that mean you can have a product of interest. You're expanding the production of something. I mean, that's the common use, right? I don't know where we are in terms of legislation. I think and I didn't speak about this area of research, but members in my team have looked at non-GMO methods of basically targeting genes for enhancing production, enhancing life, and that's a science called epigenetics. So there's some actually relatively common dietary chemicals that you can add, and some of these expression studies have been done where you can actually see. There's one study we published in 2022. Again, influencing the flavour of yeasts basically in wines. It's been quite interesting. The impact on longevity potentially has importance for a wide range of agricultural products, I would say, because, as you said, you've got a microbe with great properties. It's worthless if it doesn't survive. And so some of these non-genetically these non-genetic modifications, just by basically adding things, and the changes are not absolutely long term, but they're long enough. They're long enough. So I don't know if that answers your question, but some progress is being made. Some of it is based in, I would say, legislation. I don't know where we are with the GM. Do you, Sharon?
Well, with GMO regulation, has it made progress?
Yeah, from the agricultural point of view, I understand that we cannot use genetically modified microbes in the field, so they are all treated as a restricted organism. So we are not allowed to work with that. We can work in science, in development, in research, but not for final application yet.
George,